Hi again. In this video, I wanted to go over the organelles. And so in our notes, starting on page 9, uh, I go through the different types of organelles that could be in a cell. So I'm going to run through that in this video. So taking a look at a cell, the organelles, of course, are just all of these structures that they have color-coded inside of the cell, including the nucleus. In this video, I want to go over the organelles except for the nucleus. The nucleus is our focus next week in class. And so we're going to try to get everything else down other than the nucleus this week. And so these organelles are inside of a watery solution called cytoplasm. And so cytoplasm is just the intracellular fluid. That's another name for it. And so taking a look at our notes, the cytoplasm is also called intracellular fluid. And it's basically watery, like I said. That watery background of the cytoplasm is known as cytosol. So that's the solvent of cytoplasm. And the solutes that are in the cytoplasm include proteins. These could be enzymes that catalyze reactions. These proteins are produced by an organelle, which we'll talk about, and other things depending on the type of cell. So for instance, in skin cells, they contain the protein keratin, and that would be an example of an inclusion, something that was produced by the cell that now occupies, the inside of the cell. So I have some videos posted here. I'd like you to look at those. But I think what I'm going to do in this video is I'm just going to go down the list of the notes and talk about each one of these organelles one by one. So mitochondria, very important organelle. Most cell types, all cell types will have mitochondria, and human cell types. And in this picture from our book, you can see they're drawing the mitochondria orange. Of course, they're looking very large and compared to what they would be in a cell. But taking a look at mitochondria in another picture, just a single picture. Let's take a look at this picture. Here they're showing us mitochondria, and you can see that the mitochondria is a membranous organelle. In other words, that the outside of this organelle is bounded by a membrane that has the same, structure basically as the plasma membrane that we went over last week. So mitochondria have two membranes. There's an outer mitochondrial membrane, which they're drawing in dark orange, and that's kind of smooth. And then there's an inner mitochondrial membrane that you can see is convoluted. And so all those little folds in the membrane are called cristae. Okay, so outer mitochondrial membrane, inner mitochondrial membrane, the space in between those two membranes is called the intermembrane space, which we'll talk about later on, not really too much today. And then this area inside of the mitochondria, it's known as the matrix. And that's where the reactions of Krebs cycle occur, which some of you might be familiar with. So taking another look at a picture of mitochondria. Here's the one from our book. You can see the outer mitochondrial membrane, the inner mitochondrial membrane thrown into those folds, the cristae. You can see the enzymes embedded in that inner mitochondrial membrane because those are needed for Krebs cycle. So mitochondria, the purpose of mitochondria, as you might know, is to produce a molecule called ATP, adenosine triphosphate. And mitochondria can't just make, ATP magically. You need ingredients to make the ATP with, right? And so what we make ATP with are two things. One is a nutrient source. A lot of times in courses, people talk about sugar. So glucose would be a great example here, but also the mitochondria can use protein and they can use fat. So sugar, protein, or fat, mitochondria can use it all, plus oxygen. Mitochondria require oxygen in order to make this molecule ATP. So I'm going back to chapter 2 here to show you this picture of ATP, adenosine triphosphate. So the thing about ATP, which makes it so useful to our cells, is this phosphate tail. So you can see here's one phosphate, here's the second, here's the third phosphate on this whole molecule ATP. And do you see how it dangles off as a tail? So that's unfavorable in the cell. And so there's going to be lots of different reactants that want to interact with ATP. And the way that this works, you can see the squiggly line that they're showing in our book indicating a high energy bond. The way that this works is that different substances will be able to cleave a phosphate off. And when it cleaves the phosphate off, that's what's going to release energy. So that provides energy to the cell. and we can use that energy to do other work. It will also leave you a single phosphate, right, because you cut that off, and the ATP molecule. Now there's only two phosphates, and we call that ADP, adenosine diphosphate, which still has some energy left in it, because you could take that ADP, and you see that high energy bond, we could cleave another phosphate off. That would leave you two free phosphates in the cytoplasm, which can be used to do work, and that would also release more energy that we could use to do work. And now once you cleave another phosphate off of adenosine diphosphate, the two phosphates, that gives you adenosine monophosphate, AMP. And that base structure, the AMP, usually will turn into a ring structure, which people indicate with the small c in front of the AMP, cyclic AMP, and that doesn't have any more energy left in it. So the highest energy molecule is ATP, right, because we can cleave off two phosphates off of that, and ultimately you could, just break that down into cyclic AMP where you've removed all of the energy. But that is the point of mitochondria. So mitochondria is the organelle that can produce energy using nutrients and oxygen. And the other thing that I'll say about mitochondria, I'm going to go back to this other picture, which is kind of cool, is that mitochondria is an organelle other than the nucleus that contains DNA. And you might have heard about this, mitochondrial DNA. Here they're indicating it with that purple circle. So mitochondria contain their own DNA, which means that mitochondria replicate. It's theorized that mitochondria used to be independent structures, independent of our cells, and they took on a symbiotic relationship with our cells, from an evolutionary standpoint. So when we stress our cells, we'll talk about this more in the muscular system. Say if you go anaerobic and you stress your cells, what you'll do if you run out of energy, if you do go anaerobic, that's going to stress the mitochondria, and the mitochondria are going to replicate themselves. And that's part of physical training, so that the next time you do that exercise, you're going to be better able to make more energy because your mitochondria are replicated. So that's a little story about mitochondria for you. In our notes, I mentioned that it's a membranous organelle, that it has its own DNA, therefore it can replicate. Mitochondria use nutrients from the blood. We talked about that. Also uses oxygen. You might want to add that into your notes. I didn't mention that, but mitochondria require oxygen to make this molecule ATP. And that has potential energy stored within that phosphate tail, like I showed you, that undergo reactions to give energy. And the reactions that occur inside of the mitochondrial matrix, those are called Krebs cycle, are what result in the production of ATP. So I hope that makes sense. Let me know if you have any questions. Next one on our list is a ribosome, and the ribosome is strongly related to the endoplasmic reticulum and the Golgi apparatus. So let's talk about these as a group. So instead of learning everything one by one, let's talk about how these three things are related. So a ribosome is a non-membranous organelle. As a matter of fact, I would think of the ribosome just as a macromolecule. It has two subunits. I'll show you a picture of this. And those two subunits contain ribosomal RNA and protein. So the two subunits associate together to do one thing. They produce protein. This is the only organelle in the cell that produces protein, which can be confusing when you're first learning this, because the ER and the Golgi have to do with modifying the protein that's produced by the ribosome. So just keep this in mind, and we will talk about ribosomes again next week. Okay, but they're the only organelles that make protein, and you find them all over the place in the cell. The DNA makes ribosomes, so we have many, many, many ribosomes, and the DNA can always make more ribosomes. And you can find ribosomes in two spots in our cell. They're either floating around in the cytoplasm free, those are free ribosomes, or they're fixed, meaning that they're attached to the ER, and that's the relationship to the ER that I'll talk about next. So taking a look at a cell. So in the cell, you might wonder, like where are the ribosomes in this cell? So if you see these tiny little red dots that are attached to this blue thing, or I see some tiny little red dots out here, those are the ribosomes. So like I said, it's just a macromolecule. It's very, very small. So taking a look at just a diagram, this is from a different book, but this is showing us the ribosome. You can see how it has two subunits that interlock together. And if you know anything about protein production, the messenger RNA is going to get fed right in between those two subunits and get translated by the ribosome into a protein. So we were saying that ribosomes could exist free-floating in the cytoplasm or attached to the ER. So in this picture, this is a simple picture that's showing us some endoplasmic reticulum, and here are these dots on the surface of the ER. That would be ribosomes. Those are ribosomes. So when you have ribosomes attached to the ER, that's what's known as rough ER, and it has to do with the texture of the ER. So imagine if you ran your finger across the surface of the ER, how it would be bumpy, right? And that would be the rough ER. The rough ER has ribosomes attached, compared to the smooth ER does not have ribosomes attached. So just taking a look at another picture of, this is from our book, of the rough ER and smooth ER. In purple, this is the nucleus. In blue, this is the endoplasmic reticulum. I see that a portion of the endoplasmic reticulum has ribosomes attached. Those are the red dots. And then a portion of the endoplasmic reticulum, the ER, doesn't have ribosomes attached, and we would call that the smooth ER, again having to do with texture. If we run our finger over the surface of the smooth ER, it's not bumpy, it's smooth because there are no ribosomes there. So jumping back to the notes, as far as the ER goes, this is a membranous organelle. It's kind of like a membranous maze. You can probably tell that by the pictures that we were looking at. That membrane originates off of the nuclear membrane, and therefore it's always located close to the nucleus. There's usually lots of it in the cell, so it fills the cell somewhat. And it's just kind of like this maze, this interwoven network of channels and fluid-filled sacs called cisternae. Okay, it originates off the nuclear membrane, widely dispersed in the cell. So two main types of ER. There's the rough ER, the RER that we were talking about, that has the ribosomes attached. And because it has the ribosomes attached, protein production completely takes over, the job of the ER. I'm going to show you some pictures for this. The smooth ER, because there are no ribosomes attached, have nothing to do with protein production, and instead you're going to see the SER in cells that produce a lot of lipids. And so it doesn't have anything to do with protein production. Instead, it makes different kinds of fats like phospholipids, which make up the plasma membrane. As you know, cholesterol is made by SER, steroid hormones are made by SER. And so that's the story of the SER. It's very different from the RER, except it is still just those membranous channels where those reactions take place. So because this is originating off of the nuclear membrane, can you imagine all of these channels and sacs? They're interconnected. You can kind of see that better over here. Just because I can't see the point of attachment in any of these areas doesn't mean that it doesn't exist. It's just not in the plane of that cut. So to think about this a little bit further, the way that this works, I'll show you one more picture. So this is another picture from a different book where you can see the nucleus. You can see the RER kind of branching off of the nucleus, some of it rough ER, some of it smooth ER. Taking a look at this picture, now they're really zeroing in on the rough ER. These orange things are ribosomes. This blue area, that would be the ER cisterna. So a ribosome that's attached to the ER cisterna, when it produces a protein, that protein enters into the ER cisterna. So the ER does not produce the protein, but it receives the protein that was produced by the ribosome, and it can modify that protein. In other words, it can fold it into its higher structure, and it can add sugar groups. And that's the kind of work that you see going on here. Now, because that protein was produced inside of the ER, the only way to release that protein would be to package it in a membranous vesicle. So that protein is going to be packaged in a membranous vesicle, and it's destined to always be packaged in a membranous vesicle. So here's a big difference. If I had a ribosome, a free ribosome that was free in the cytoplasm, it could make a protein, and when that protein is produced, it enters into the cytoplasm, and it's destined to always stay in the cytoplasm. I hope that makes sense. It would have no means of getting out of the cell. And so it's probably an enzyme or something like that that's going to work in the cytoplasm, compared to a protein that's produced on a fixed ribosome. Because it's packaged in a membranous vesicle, this protein could be released from the cell through exocytosis. The instructions on how to make the protein, which we're going to talk about more next week, will indicate to the ribosome where it should be. And so a ribosome can flicker in between the two states. A ribosome can be fixed, but if it attached to instructions that say it should be free, it will detach from the ER. or if you had a free ribosome that attached to messenger RNA, and in that recipe it tells the ribosome it should be attached to the ER, it could dock onto the ER and produce that protein there. So I told you how the ER is affiliated with the Golgi, so let's think about that for a minute. So in this picture, oops, sorry about that. In this picture, what we're seeing is the ribosome on the surface, the protein is being produced on the RER, and then the protein is being released by exocytosis from the ER, and that protein is then going to go to the Golgi apparatus. So the Golgi apparatus is a little bit like a chain reaction. Once the ER finishes its work with how it can modify the protein, it's packaged in a membranous vesicle, and the Golgi apparatus is kind of like a membranous stack of pancakes. These stacks, the pancakes, so to speak, the fluid-filled pancake, that's also called a cisternae or cisterna, singular. So the protein enters into the Golgi, and the Golgi can further modify the protein. So taking a look at this picture from our book, here they're showing us the Golgi. What would happen is the bottom of this stack, it's called the cis face, will receive the membranous vesicles that have the protein in it coming from the ER, modify it, and then coming off at the transface of the Golgi, the protein in their final form will be packaged in a membranous vesicle. So those, I would think of the Golgi as putting like the finishing touches on the protein. So just going back to the notes, the Golgi is the membranous stack of fluid-filled sacs called the cisternae. It's close to the RER, and what it does is it receives proteins from the RER, and then it further refines those proteins. You know, it can do that by adding, changing sugars. The way I think of it, like I said, putting the finishing touches, because when the protein leaves the Golgi, it's done. It's not going to go on to like another organelle for further refinement. So because these proteins that leave the Golgi apparatus are final, it's one of three things will happen, pardon me. I mentioned this, it could be secreted from the cell using exocytosis. So maybe this is like insulin being produced by a pancreatic cell, and now it's going to be released from the cell through exocytosis. It could become integrated as part of the plasma membrane. So as part of the refinement of the protein, that protein could get lodged within the membrane of the Golgi, and so it doesn't really get secreted from the cell. Instead, it becomes part of the cell membrane, and that's how we can put receptors and carriers and channels up on the plasma membrane. Or it could even be an enzyme, and then this membranous vesicle is retained within the cell, and it's a organelle. And so those enzymes are catalyzing a reaction, and that reaction is doing work in the cell. And actually the next two organelles that I have up on our list, the lysosome and the peroxisome, are related in that they are just membranous vesicles that were produced by the Golgi that contain certain types of enzymes that are going to do a certain job in the cell. So the lysosomes, these are very common in cells. The lysosomes contain enzymes that you can think of as kind of like the garbage can of the cell. They're hydrolytic enzymes where anything that's fused with the lysosome is going to break it down. So inside of the lysosome, it's very acidic. Basically anything that enters the lysosome is degraded. Okay, so that's how we get rid of waste within the cell. Now the peroxisome, it's similar. It's a membranous vesicle, but the enzymes just do a different job. What they do is they work to get rid of a certain type of waste called an oxygen-free radical. And so an oxygen-free radical is just a molecule that contains oxygen, and it has an unpaired electron in its outer shell. So it's very, very reactive. And because it's very reactive, it's implicated in aging, disease, inflammation, all kinds of things we don't want anything to do with, right? And so our cell has a mechanism to get rid of this type of damaging reactive waste, and that is the peroxisome. So it contains enzymes that will actually take the oxygen-free radical, turn it into hydrogen peroxide, and then get rid of the hydrogen peroxide. So they help rid our cell of that type of waste. Now the next little bit of organelles that I have in the notes, the cytoskeleton, the centriole, and the centrosome, those three are related. So those three are related in that these are all types of protein tubules, and they're playing a structural role in the cell. In other words, it's going to be like a physical structural role. So the cytoskeleton is protein tubules, all different types, micro filaments, intermediate filaments, thick filaments. You can see the small, medium, and large, basically filaments. Filament being like a solid protein fiber compared to a microtubule, also very common, but you can hear in the name too, that's a protein that's hollow like a straw. And so all of those proteins can create like an infrastructure within the cell that gives the cell a shape. So I want to take a look at that. So here's a picture. These orange kind of like spider web would be microfilaments, microtubules making up the cytoskeleton of the cell. And so just like our body has a skeleton made of bones that give us structure, the cell has a cytoskeleton made of protein, which gives its distinct shape. But more than that, what it does, believe it or not, this goes beyond the scope of our course, but these protein filaments act as like railroad tracks too, like membranous vesicles. And so say you had a membranous vesicle coming off of the RER and it wants to go to the Golgi, how's it going to go to the Golgi? Well, one way that it could go to the Golgi is that membranous vesicle uses this kind of like a railroad track, like I was saying. And so this has a lot to do with structure and movement, transportation within the cell. And so if you take a look at our book, they do go through the different types of filaments. Okay, so microfilament, intermediate filament, thick filament we'll talk about in muscle. So these play different roles within the cell. Microtubule, we talk a lot about microtubules in the class. It's made out of a protein called tubulin. But you see how that protein, which is a spherical protein, gets assembled so that it forms kind of like a hollow straw. And so those are very prevalent too. So that's the cytoskeleton. And looking back at our notes, microtubules make up something called a centriole. And a centriole is like a distinct kind of arrangement, like a hollow barrel of microtubules that form a structural anchor in the cell. So let me show you what that looks like. So here's a picture that shows what's called a centrosome. Okay, so the centrosome is what you call when you have two centrioles existing at a right angle. So here's one centriole and here's the other centriole. So do you see how one is running, more vertical and one is running more horizontal? So that's what we call a centrosome. So centrosome, that's common in the cell because the centrosome is what we're going to use to anchor the mitotic spindle for cell division. So the centrosome is made up of two centrioles. So a centriole would be one of these, which is this like little barrel type of arrangement, like I was describing. And you can see there's this particular arrangement where you get the three microtubules and they radiate out. Okay, so all centrioles have this exact shape, and then it's hollow in the middle. And it's a very strong structural anchor. Microtubules can make more microtubule, and so the spindle fibers that we'll talk about next week that form the mitotic spindle actually grow off of the centrioles. And another anchoring role that centrioles have is to anchor like cilia onto the cell surface. So, and I'll talk about cilia next up. But here, cilia are just little extensions from the cell surface. And so if we look here right at the base of the cilium, what we would see, as they're showing us in this picture, here's the cilium. And then what's right at the base, there's the cell surface anchoring that cilium to the cell. Well, there's the centriole we just talked about. So centrioles are like strong anchor points in the cell. So we looked at centriole. Centrosome is just the two centrioles that exist together and form the base of the mitotic spindle. And so moving on to the last thing I want to talk about as organelles, and these aren't really organelles, but these are specializations that can come off of the cell surface. Okay, so there are other cellular extensions. Take a look at that in the book for yourself. But the three types that we're concerned with, cilia, I mentioned those. Whenever a cell has cilia, there's going to be lots of cilia. So that's why I put this kind of analogy. It's going to be like blades of grass in the lawn. There's not just one cilium. The whole surface of the cell will be coated with cilia. And the cilia aren't just filled with cytoplasm, they're filled with cytoskeleton. And so taking a look back at the picture, here's the cilium. Do you see how it has this particular arrangement of microtubules inside? This is what's called a 9+2 arrangement of microtubules, where you basically have that kind of like outside, like the centriole, and those two microtubules in the middle. What that infrastructure is going to do for a cilium is it allows it to be a modal structure. So I'm going to go back to this picture, and I'm also going to try to describe this with my hands if this makes any sense. So say like my hands are two cilia. What they do is they have a coordinated type action where they work together, say like windshield wipers, even though there's going to be like not two of these, there's going to be like 2 million of these coating the cell surface to sweep something across the surface of the cell. Okay, and so cilia is a modal process, but it doesn't move the cell. It moves something across the surface of the cell. So for instance, in this picture, they're showing us, here's a layer of mucus. And so as an example, the cells that line our windpipe or our trachea are ciliated cells. So you can see these cilia work in a coordinated type of action to actually move this mucus up towards our throat. Another example, the lining of the uterine tube is ciliated. You might know that the oocyte, the egg cell, it's not modal. It doesn't move. So how does it make it through the uterine tube from the ovary to the uterus? Answer, because cilia move in a coordinated type way to move that cell down the uterine tube. So that's cilia. That's a modal process. Let's compare that to, I'm going to jump to another picture here, then I'll go back to the notes, microvilli. Microvilli are non-modal. Okay, microvilli, sure, they have a cytoskeleton on the inside, but they don't move. So these are more or less just, like floppy, fleshy extensions of the cell surface, if that makes sense. They don't move in any type of particular fashion. What they do is they increase the surface area through which the cell has to do work. So for instance, if you have cells with microvilli, you can see it's coated with microvilli. Can you imagine if that cell surface was, say, like, I'm going to say that's like a foot long, or if it contains cilia, and it's also a foot long, can you imagine that if I cut that little piece of membrane off and stretched it straight, that's going to be several times the length of that area. And so that's what I mean by increasing surface area. The microvilli give a lot more area to do the job, say, for instance, absorption of nutrients. Cells that contain microvilli line the small intestine. So if you had nutrients out here, I have more space to bring those nutrients into the bloodstream, to absorb those nutrients into the bloodstream. And so those two kind of look similar, although they work totally different. So I'm going to jump back to the notes. So cilia, they're modal, and microvilli are non-modal, but they both cover the cell. Now, flagella is a modal process, but if we're talking about a human cell, which in this class all we care about is human cells, you're looking at a single long tail-like extension. And there's only one human cell that contains flagellum, and that's a sperm cell. So if you're familiar with the tail of a sperm cell, what that flagellum does is it actually propels the cell. The flagellum will have an infrastructure like the cilia, so it has that 9+2 microtubule arrangement anchored to the sperm cell by the basal body or the centriole, and then it moves so that the cell actually moves to the site of fertilization. So a flagellum, singular, is a modal process also. So that is the short story for that part of the notes, and next week we're going to wrap up this unit by talking about the nucleus. Okay, I'll see you again soon.